All right. Hi. So thank you so much for giving me this opportunity to speak today about Alfy. I'm so very excited to be able to present to such a learned audience of Huntington's disease research. It's something I rarely get to do lately. So I've worked on Huntington's disease right throughout my entire research career, and I entered the field at a time when it was just really exciting. This disease that was characterized for 100 plus years largely by neurological and neuropathological features was now being, thrust into like kind of molecular mechanisms by, of course, this, absolutely landmark study by the Huntington's Disease Collaborative Research Group, right, who identified the gene and causative mutation that led to this adult onset neurodegenerative disease. Now, of course, as we, identify the mutation underlying the disease, it was quickly learned that this caused, the protein to change its ability to be in the cell. And that major change was that it started to aggregate. And this was perhaps a rediscovery, say, of Roizen's work in the 70s, but of course, the polyglutamine expansion mutation that was identified ended up causing this protein to be highly aggregation prone from in vitro to in vivo. Now, when I started, the adult onset neurodegeneration field was really starting to look at protein accumulation, across adult onset neurodegenerative diseases, raising questions as to whether or not it's a common theme for pathogenesis. And the discovery that the HD causing mutation caused Huntington protein to aggregate I felt like thrust HD research right up there with the less tractable but more prevalent, sporadic disorders like Parkinson's disease. And Huntington's disease really allowed us to kind of gain a window into understanding the aging CNS and health. Now, of course, though, the focus is Huntington's disease, and the identification of the gene and mutation causing HD had such an impact to patient and patient families, because now we can use approaches to identify those potential patients that will have this kind of devastating outcome. And so, of course, in that excitement was also the reality of trying to frame, our questions of understanding, when this mutation was expressed, what were the earliest events that led to symptoms? And, can we find a kind of targetable approach by which we could potentially treat HD? And so it was kind of in this where I entered the field. And when I was with Renee Hen and Jose Lucas, we were inspired by the work, the pioneering work by Jill Bates, who showed that expression of mutant huntingtin in a mouse was sufficient to drive symptoms. And, so we, like the many that followed her, also created a mouse model to try to understand the interaction between expression of mutant huntingtin and symptomatic onset. And like many, what we saw was under gene-on conditions where mutant huntingtin is expressed, at some point the mouse would become sick and they would show motoric symptoms. On a neuropathological level, they also showed the aggregated mutant huntingtin protein abnormally accumulating throughout the cell. Now, our mouse model, we used a genetic switch, and what this allowed us to do was to get insight into the relationship between expression of this potential toxin and the disease outcomes. And so what we did, of course, was shut off gene expression and found that at that time in this mouse model, you could have symptomatic reversal. And so this suggested that there was a period of time when you could take a step back, that continuous expression of mutant huntingtin was necessary to drive the symptoms. Now, what blew my mind about these findings was on the neuropathological level. And I still remember to this day kind of looking through the slides and being all freaked out because I thought I mislabeled things and it was really a big confusion. So, the aggregates went away. Now, this blew my mind largely because at the time, when we were studying aggregation, it was understood that the polyglutamine expansion led to the formation of very, very stable structures. Something like formic acid was necessary to break them down. So the idea of simply eliminating expression of mutant huntingtin to reveal the ability of cells in the adult brain to eliminate these structures really caused a lot of wonderment for me. And I think this kind of scientific question, the interest I had in this scientific question really crystallized when I was asked a very simple question when I first presented these findings. And this was by the late Max Perutz, who asked, Dr. Yamamoto kind of thing. I wasn't a doctor yet, so I was kind of embarrassed by that. Where did the aggregates go? A simple enough question. I, of course, had no friggin' idea where they went. And, it was really at that moment I resolved to try to figure it out. Of course, it actually didn't take very long, not because I was particularly fast, but all I really had to do was have some time to read. And that came when I was writing up my dissertation. And I found work by Ron Capito. And in his agrosome hypothesis, he proposed that there was a pathway in the cell that could degrade these structures. And that pathway is known as macroautophagy. So macroautophagy is the means by which cytosolic proteins are eliminated by the lysosome. And it's accomplished by encapsulating cargo that into a double membrane structure. They're denoted in green, known as the autophagosome. And that autophagosome can now fuse with the lysosome, and thereby allowing for that low pH to pull apart the aggregates and ultimately let the proteases break down the inclusion. So to kind of test Capito's hypothesis, we decided to do an unbiased screen, actually, and I did this as a postdoc in the Rothman lab. So to do this, we reduced the mouse model, this inducible expression of mutant huntingtin, into individual cells. We used a stable HeLa cell line. And here we express the fluorescently tagged mutant huntingtin. The fluorescence is there shown in cyan. And you can see over as the polyglutamine expansion number increases, the probability by which you can see those like little dots on these aggregated structures increased, showing that kind of Q-length dependent increase in aggregation. Now, if you look in panel C, you could also see that upon shutting off expression of mutant huntingtin in these cell lines, also revealed this ability of cells to eliminate these aggregated structures. So this was a common feature across different cell types. It wasn't necessarily something unique to cells of the adult brain. So using this cell line, we embarked on a unbiased genetic screen. We were using siRNAs and cDNAs, and it was an image-based screen. So there under 3, you can see kind of these panels. In blue are nuclei, and the green starry-like things are the aggregates. And so if you look to the left, you can see if you add doxycycline, shut off gene expression, those aggregates go away. But then sometimes what would happen is we would transfect an siRNA that depleted a critical protein in this pathway, and that would impede clearance, as shown on the right, and we could quantify this. And using this approach, we soon identified various genes that were implicated in the formation of the autophagosome itself. So at the time that we really got this work going, 2 papers published. First, and in fairness, Ron Capito actually published twice showing that macroautophagy can indeed clear aggregated structures. And I believe it was aggregated mutant huntingtin that he showed. Fair enough. But it was good to see that our unbiased screen was basically working and we were able to confirm his results. The second paper actually made us take pause a bit because we were a bit confused by what we were seeing via what was being reported. So the second paper was by David Rubenstein, who presented exciting results suggesting that increasing macroautophagy in the brain of a mouse model of Huntington's disease led to clearance of aggregates and ultimately protection. So this was very exciting, but what gave us pause was the means by which macroautophagy was activated, and that was by inhibition of mTOR using rapamycin, or maybe it was a rapalog. Now, there was nothing wrong with that, actually. In fact, macroautophagy was discovered under conditions of prolonged starvation when mTOR was inhibited for a prolonged period of time. What gave us pause was what we were seeing. What we were seeing in this clearance assay was the clearance of aggregated proteins in these cells that were under full serum. I mean, massive quantities of insulin, massive quantities of amino acid, the absolute antithesis of starvation. In fact, in the adult brain work that we had already done, also, all we did was shut off expression of mutant Huntington. So what were we seeing? Were we actually seeing autophagy inhibition? Was autophagy actually responsible for clearing the aggregates? Or was the inhibition kind of messing with protein homeostasis overall and thereby leading to the accumulation of mutant protein? So to kind of we took a step back to try to really understand whether or not macroautophagy was playing a role. And so, we followed that old adage of seeing is believing. And the idea was, well, if aggregated proteins are turned over by macroautophagy, we should see aggregates inside an autophagosome. And so that's something that we really wanted to pursue. Now, we used immunofluorescent-based approaches. That turned out to be sort of promising, but not really definitive. And so we decided to kind of turn to the gold standard and do ultrastructure. And this turned out to be really challenging. But finally, with Andreas Breck at the University of Oslo, we made some headway. So here is some immunogold labeling of the mutant Huntington cell lines. And we saw two different structures. One structure, which is to the left, was kind of this amorphous, large, more than 1 micron size structure. And the other structure to the right was this kind of electron-dense structure, 1 micron and less, that seemed to be delimited by a double membrane, as indicated by the two arrows. And so, we were like, okay, this sort of fulfills one of the morphological criteria of Autophagosome. It's a cargo seemingly captured by a multilamellar structure. But what gave us pause was really the fact that we were missing a second key morphological feature, and that is beautifully illustrated here. So here is an image taken by Eva-Lisa Escalane. It's my favorite picture of an autophagosome, if anyone has a favorite picture of an autophagosome. Anyway, you see a multilamellar structure labeled here AVI that's encapsulating a mitochondrion. You can beautifully see the cristae. But around the mitochondrion in white is is the second morphological feature of an autophagosome, and that is bulk cytosol. Right? We had no bulk cytosol in our structure, just cargo and the double membrane. So we weren't quite seeing what was defined at the time as an autophagosome. But instead what we saw was this structure. Here you can see this electron-dense structure encapsulated by a double membrane. This is an image taken by Dan Kleonsky's group. And Dan was studying a pathway known as cytoplasm to vacuole targeting. It's a pathway in yeast that targets oligomeric structures with an autophagosome-like membrane to the yeast vacuole. And the structure that's seen here is what was known as the CVT, or cytoplasm to vacuole targeting vesicle. And as you can see, it looks very much like the structure that we see, an electron-dense structure with a double membrane. One of the defining features of CVT is that it occurs in full nutrient conditions. And this is what distinguished it from the very related cousin pathway, macroautophagy. So at the time we thought, hey, we discovered CVT in mammalian cells, but at the first autophagy meeting I attended, I was told, no, there's no CVT in mammals. So we were kind of wrong. So we were still kind of befuddled as to what it was that we were looking at. But that clarified itself soon enough. So as we learned more and more molecularly about the players involved in the formation of the autophagosome membrane, it soon became very clear that the CVT vesicle was indeed an autophagosome membrane. It was one and the same with that beautiful structure that Eva-Lisa took. The only difference was that bulk cytosol was excluded, And the manner by which that was excluded was through adapter proteins. So here in the schematic in orange and green, you can see labeled ATG19 and ATG11 are two key adapter proteins that play a role in CVT. And these adapter proteins basically scaffold the growing autophagosome membrane. They're labeled with ATG5-1216, ATG21, and PI3P. To allow it to tightly wrap around the aggregate in yellow and avoid and prevent bulk cytosol from coming out. And since kind of this original understanding, it came to be, that actually this pathway that Dan was working on, CVT, was not some esoteric yeast pathway, but it was actually the paradigm for what is known as adapter protein mediated macroautophagy, also known as selective macroautophagy. What I would argue is probably the dominant facet of a feature of macroautophagy that's occurring in our cells even now, where every single organelle in the cell is probably in an adapter protein dependent manner being turned over by macroautophagy under full nutrient conditions. And as you can see, in addition to a variety of organelles that have already been characterized to be degraded this way, other structures from microbes, as well as stress granules, and of course my favorite, proteinaceous inclusions, aggregates, are degraded this way. So one of the things that we had also come across in our screen was actually the adapter protein that was responsible for targeting aggregated proteins to the inclusion, and that is known as the autophagy-linked 5-domain containing protein. This protein was first characterized by Anne Simonson when she was a postdoc with Harold Stenmark, and since then she and I have continuously collaborated to understand how Alfy may potentially play a role in vivo for aggregate turnover. So in essence, it's a 380 kilodalton mammoth scaffolding protein, and at the C-terminus end, it has a series of protein-protein and protein-lipid interaction motifs. And in essence, kind of modeled against CVT, it helps to scaffold the aggregate with the growing autophagosome membrane as we model it to form that CVT vesicle-like structure. So the primary data looked like this. Again, it was in our screen. You can see that the knockdown of Alfy using 2 different siRNAs prevented that clearance of aggregated proteins. And so while we were very excited to identify this structure, one of the features that we also wanted to understand was how specific was Alfy for the turnover of aggregated protein. And the reason we were interested in that, of course, had to do with disease context. If aggregated structures were not pathogenic, perhaps it was the soluble form. And so in order to be able to distinguish between the two, or we wanted to know if we could distinguish between the two, we wanted to see if Alfy impacted here beyond the aggregates, also the soluble protein. So to do that, we turn to the filter trap assay. And this is a bit complicated image, but if you could focus to the right under Q103, and the circular dots that you see is the filter trap, capturing insoluble mutant huntingtin. And as you can see under expression, under full expression, you have a nice dark dot. And if you shut off gene expression with DOX, that dot gets lighter. If you shut off gene expression, but also deplete Alfy, the dot persists, right? And the aggregated proteins are still there. To look at this detergent soluble protein, we then turn to SDS page. And here you can see expression right above the filter trap. It's labeled SOL for soluble. And if you give doxycycline, shut off gene expression, the amount of soluble protein goes down. Of course, we make no more mutant huntingtin, right? And it gets cleared. But if we deplete Alfy now and shut off gene expression, it continues to clear. So it's really only the aggregates that persist upon depleting Alfy. So this suggested to us some degree of specificity. So that was all in cell lines, and so we wanted to know if this also worked in the adult brain. And so this was work done by Leora Fox in the lab, a real mammoth undertaking, She first had to recreate the TET regulatable HD mouse model, which unfortunately had a very limited expression, but suffice it to say it was sufficient for us to understand whether or not the abnormally accumulated mutant huntingtin could still clear, as shown here, upon shutting off mutant huntingtin expression. To try to understand Alfy dependence, we didn't want to do virus or anything like that. We want a kind of uniform knockout. So Leora created an inducible knockout of Alfy, where she could temporally regulate when you can make a gene knockout. She then took the next 2 years crossing the two animals together to make sure all of the genetic pieces were on one mouse, and then asked the question, if I tried to clear aggregated proteins in the absence of Alfy, what would happen? And lo and behold, what she found was that the mutant huntingtin, despite shutting off expression of new mutant huntingtin, could no longer be cleared. So this suggested that just like how we saw clearance could go from brain to HeLa cells, the HeLa cells were being predictive to kind of give us insight into the molecular mechanisms involved for aggregate turnover, and Alfy was a common feature between these two cell lines. We also wanted to, of course, to bring it back more to disease relevance and patient materials. For this, we collaborated with Andrew Yu's group, who uses a microRNA-based approach to directly convert dermal fibroblasts into medium spiny-like neurons. And we chose this because he had just such wonderfully remarkable findings where he can, unlike iPSC-derived neurons, which was another option we were pursuing, in direct conversion, one of the things that he could see was aggregation of mutant huntingtin even with low polyglutamine length. So here we did it with 46Q, but for the study we did also 40Q and 47Q. And even with these smaller polyglutamine lengths, we saw aggregation. And what was quite exciting was that depletion of Alfy augmented aggregation, suggesting that by diminishing the amount of Alfy available to the cell, we were slowing the ability of the cell to turn over inclusions and thereby causing more to accumulate. So now it looks like we have a protein that plays a critical role in the turnover of just the aggregated form of mutant huntingtin. So we were now in the position to try to understand whether or not impacting aggregation had any impact on a symptomatic outcome. So to do this, Leora first did the loss of function study. She did this by depleting Alfy by 50% in back HD mice. We did this because 50% depletion led to no phenotype, no detectable phenotype under a wild type background. And so we were really concerned of kind of like a sick getting sicker outcome. And so using these mice, what she was able to find was that the depletion of Alfy in the back HD mice really accelerated the aggregation phenotype, as you can see below. Some of you might be familiar. The back HD is actually a very low aggregation model, but when we depleted Alfy, they were quite straightforward to find. Behaviorally, so what happens? Here I show above accelerated rotarod. You can see that the back HD mice in blue manifest at about 5 months of age. And then below you see distance traveled in the open field, and the back HD mice show a hypolocomotor phenotype at 12 months of age. Well, when aggregation was accelerated upon depletion of Alfy, so did the onset of the motoric symptoms. So first at 3 months of age already do you see changes on the accelerated rotarod, and at 9 months a hypolocomotor phenotype. So here, what seems to happen is that the loss of function, during the loss of function, you have increased aggregation, right? You slow aggregate turnover, increased aggregation, and then you accelerate the onset of motor phenotype. Given these outcomes, we wanted to make sure that our previous findings about soluble protein was still relevant in the adult brain. And this is just shown with 2 different Western blots, the soluble on the left, the detergent insoluble, the urea soluble on the right. And I hope you can appreciate how when you take the back HD or BHD, heterozygous mice in pink, the soluble proteins are indistinguishable from back HD, but the aggregated protein is significantly higher, again indicating that detergent soluble levels are not affected by Alfy, only the detergent insoluble aggregated protein is. So again, here we, by augmenting aggregation, symptoms seem to be accelerated. So the next question, of course, was a gain of function, right? If you deplete Alfy levels, maybe the mice are sick, we don't know. And so gain of function experiments could potentially show us whether or not we can A, accelerate aggregate turnover by increasing Alfy levels, and B, whether or not that has any impact on symptomatic outcome. We had some idea that increasing Alfy levels could enhance aggregate turnover, and that Alfy was rate limiting in this pathway. And while I don't have time to really explain this figure in its entirety, if you please focus just on the gray bars, this will kind of summarize what it is that I would like to tell you. So if you look at the number of cells with aggregates, and you overexpress Alfy for like a couple of days, we can already see a significant depletion of the total number of aggregates inside the dish. And so, we thought, well, that's enough for us to go check. And so Catherine Croce then was the brave graduate student to take on the next behemoth mouse study, and this time create an overexpression model where Alfy is overexpressed through knock-in on the Rosa 26 locus, and then crossed it to two different mouse models of Huntington's disease. The N171 model, the N171 model, as well as the CAG140 knock-in model. So both of these models are high aggregation models, as is well known. Here's images of some staining, the N171 at 5 months of age, the CAG140 at 6 months of age. So what happens when we overexpress Alfy? So Alfy overexpression here is from the beginning of time, as it were. And as you can see, we far exceeded what we expected to see, where we see really few inclusions at this time, a very dramatic rescue. Again, consistent with the HeLa cells, has suggested that Alfy's rate limiting, augmenting Alfy, increase the rate of turnover of Alfy by macroautophagy. So the million-dollar question, what happens to symptoms? So for simplicity's sake, on top I show accelerated rotarod with the N171 model, and below I show open field locomotion of the CAG140 model. For both models, you can see in red the phenotype onsets, 2 months of age for the N171 rotarod phenotype, and at 6 months of age, a hypolocomotor phenotype in the CAG140 mice. Now, what happens if you overexpress Alfy? Again, aggregation is significantly diminished, and you can see a dramatic delay in the onset of these symptoms. So here you can see above in green how the mice just don't fall off the rod anymore, and in the open field, the mice now run similarly to control. So taken together, we now can see that by modulating Alfy levels, we can modify the age of onset of the motoric symptoms in these mouse models. And this seems to be directly connected to the aggregation phenotype, suggesting that protein accumulation in at least these Huntington's disease models seems to interfere with circuitry function and causing the manifestation of these symptoms. Okay, so that's everything. So in essence, what we found was that, Alfy is this potential modifier of the aggregation status, and this can potentially have a profound impact on the motoric symptoms that we see in the mice. As you can see from the kind of hot off the presses data, we're well on our way to try to understand whether or not this has a significant impact in other proteinopathies as well. So here are my acknowledgments. This work, of course, would not be possible without everyone, basically, that's been shown on the slide. I hopefully mentioned everyone whose work I directly showed, but the people in yellow are the ones that directly influenced or impacted this work by blood, sweat, and tears, or with cash. But of course, especially in terms of the support, all of the support was very important in one way or another to help this work happen. And so with that, I'm done. Thank you very much for your attention.